Okay, so this video is going to put together what we learned about restriction enzymes and DNA ligases. This one directly links to a dot point from the study of design that says the use of recombinant plasmids as vectors to transform bacterial cells. So I'm going to put all of those words into this video as we go through, and I'll highlight them to make sure that you're aware of what is a recombinant plasmid, what is a vector, and what does it mean to transform a bacterial cell. But before we start that, we need to think about insulin again. So I did say that insulin is a really good example for multiple reasons in the course. So insulin is a hormone. Insulin is a protein. So you can automatically see that we are talking about signaling molecules. We're talking about protein manufacture. So you could link that back to DNA as well. So insulin links in with a lot of different parts of the course, and now it's going to link in with our biotechnology. So this is one of the very first types of genetic engineering that we've done as a society, and it's been used since the 80s. We're very comfortable with it, and it's where we manufacture a lot of the insulin so that diabetics require. So what is insulin? Just a little refresher. Insulin is a hormone, a peptide hormone that is produced in your pancreas. So I'm just going to reconnect this. There we go. So insulin is a hormone that is produced in your pancreas. Sorry, guys. I'm just going to leave that up the top there because this is now the third time I've tried to record this video. So here is your pancreas. Over here it sits kind of underneath your liver, right in front of your stomach. It's responsible for producing insulin and another hormone called glucagon, and they regulate your blood sugar levels. It's also responsible for producing enzymes that help you digest your food. So here is the pancreas. The pancreas normally produces insulin. This here is the insulin protein in this second picture. Now, people that are diabetic have malfunctioning beta cells, type 1 diabetic, so they cannot produce insulin themselves. So when they eat a meal, their blood sugar level goes up and they can't reduce it using insulin. So back in the 20s, a very, very important and famous, sorry, not famous, but an important scientific discovery was made, and that was the use of what we call porcine insulin for diabetics. So they would extract pancreases from pigs and take out the insulin and purify it and give it to diabetics so that they would be able to lower their blood sugar levels and not get any of those hyper glucose reactions where it's not particularly great for their system. So we've got this porcine insulin being used. These guys over here, if you're wondering, these are the beta cells in the pancreas. These are the malfunctioning ones in people with type 1 diabetes, so they don't work at all. So porcine insulin was extracted from pigs, purified, and given to humans. Now, there were a few issues with that, and we're going to explore that in the next video. But we needed a new way to manufacture insulin. This is a bit slow. It's a little bit allergic. Some people would have allergies to porcine insulin, and we don't want them to have allergies to something that we're trying to get them to help with their blood sugar levels. So what we came up with was this process called bacterial transformation. So that means transforming or changing a bacterium to make it do what we want it to do. So let's have a think about what is a bacterium. So it starts off with a cell membrane. It's got a cell wall. It's got a really long, this is a really bad picture. It's got one long circular chromosome. It's got no chromosome. It's got no nucleus because it's prokaryotic, but then it also has these little guys here. Now, these little circles. So these little circles, and there's heaps of them within the bacteria, they are called a plasmid. Now, a plasmid is just a short, circular piece of DNA. Yep. We can map the base pairs easily. They reproduce independently of the bacterium, and they're easy to manipulate, easy to take out, easy to put back in. There's also the fact that there are heaps of them, heaps in each bacterial cell. So there's a whole bunch of reasons why we're going to use these plasmids, but you need to understand here is our bacterium. And feel free to pause if you want. Here is a picture of a bacterium. These are the general features that we need to understand. Most of them have a cell wall, they have a cell membrane, they have one long circular chromosome, and in addition to that, they also have these things called plasmids. And it's the plasmids that are important when we talk about bacterial transformation. So, what are we going to do with this? First thing is we've got to figure out what we're going to transform the bacteria to do. So in the case of insulin production, we're going to take out the DNA from a human cell. So this is the DNA that contains the insulin gene. So over here, you can see here is the insulin gene. And we're going to use a restriction enzyme and cut out the insulin gene. So we're going to cut out the insulin gene using this. Now, usually we leave sticky ends. Why would we want sticky ends? We want that complementary and very specific binding. So we're going to cut them out using sticky ends. In addition to that, we're also going to remove a plasmid from a bacterial cell. Okay, so this is the original plasmid here. Now, this video over here that says watch this first, I'm going to link that in the playlist, so I want you to watch it at some stage, and we will also watch it in class. So now we've got a plasmid from the DNA, from the bacterium, and I've got my little cut out piece of DNA from a human that is an insulin gene, a functioning insulin gene. So that could be my DNA because I don't have type 1 diabetes. So now what we're going to do is we're actually going to use, and this is the really important bit, we're going to use the same restriction enzyme to either cut the plasmid or cut it. So we can cut that here. This is the same restriction enzyme as before. And now we end up opening up our plasmid, and it'll have sticky ends on either end. So here we've got our open plasmid. Now, because we've used the same restriction enzyme, those sticky ends, so they insert the gene for insulin into the plasmid, because they have complementary sticky ends. Okay, so because they have complementary sticky ends, we can insert the gene for the insulin into the plasmid. Now, this is that first word that we're going to come across. So this guy here that has the insulin gene in the plasmid, this is called a recombinant plasmid, because what it has got is it's recombined. So we've cut open the plasmid, we've inserted the gene for insulin. We also use ligase to join them back together. So the ligase, I'm just going to do that in green. So ligase sticks here and here. Yeah. So that's what the ligase is used for, to stick that insulin gene into the plasmid. And now we have this thing called a recombinant plasmid. Now, what these are is these are also called vectors. Yes? So they are vectors because they are going to carry the gene from, from one organism to another. Yeah, but they're not going to necessarily need it themselves. They are just literally carrying the gene from insulin back to the bacterium. So the most important thing is to make sure you put the, sorry guys, I want you to change your notes here. So when it says plasmid, I want you to say put the recombinant plasmid with the insulin gene back into the bacterial cell. So here is the recombinant plasmid. You put it back in the bacterial cell. Now your bacteria are going to divide. You've got to remember that there are going to be multiple copies of bacteria, and now all of these bacteria, all bacterial, I'm going to call them offspring, can produce insulin. Yeah, so now we've got all these bacterial offspring, they can produce insulin. We now can extract that insulin, we can purify that insulin and use it for humans. So the process of cutting the DNA of one organism, so in this case, it's the human insulin gene that we've cut out, and inserting it into the DNA of a bacterium. That's what we call bacterial transformation. So we've transformed the bacterium to now contain the gene for human insulin. So first off, I want to know, why would we use bacteria? So I want you to pause, I want you to stop, I really want you to think, why are bacteria? We're going to come up with 3 reasons at least why we think bacteria are really useful for this, this process. So pause. Hopefully you did that and you're coming back and you're listening to my musings on this issue. So the first thing is, ethically they're fine. Honestly, no one really cares about the feelings of bacteria. We haven't got any studies to indicate that using them as a vector is going to cause ethical issues. So therefore, ethically, we've got no issues with this. Ethical issues to do with pigs and cows, however, large mammals, very intelligent organisms, not great. Another reason, they are hugely fast at reproducing. Some bacteria can reproduce and they can double every 20 minutes. So if you want to use this to your advantage, we can now make hundreds and millions of bacteria very, very quickly using this process. They are also tiny. So bacteria are tiny. Remember, in our bodies, we have 10 times as many bacterial cells as we do human cells, but we don't even see them at all. So they are very tiny. They are very easy to grow. You just give them some food in a big giant vat and they will start to divide. Then they will start to do your bidding and they'll start to produce that insulin for you. So really, really helpful to use bacteria to produce a lot of a particular protein. And in this case, that protein is the insulin protein.